Imagine your brain signals turning up so loud that every sound, every thought, and every single moment feels overwhelming. That is Fragile X. Fragile X syndrome is the most common inherited cause of intellectual disability and is caused by a mutation in the FMR1 gene on the X chromosome. About 1 in 7,000 males and 1 in 11,000 females are diagnosed. The mutation of the FMR1 gene lowers FMRP, a protein that keeps brain cell connections working properly for learning and memory. Children with Fragile X often show developmental delays, learning challenges, speech and language delay, attention problems, and hypersensitivity. There is no cure, but care is supportive. Clinically, we still need reliable brain-based markers that show a drug changing the target signal. Now picture a busy traffic intersection. FMRP is a traffic light that keeps synaptic protein making in check. Red means pause protein making, where green means make. Because the protein is absent in Fragile X, the light fails, so traffic surges. Inside our neurons, messages move through signaling pathways that decide how much protein to make. One key pathway is the ERK pathway, or ERK. Now, how is ERK actually activated? Signals outside the cell hit a receptor on the cell surface. That message is sent to MEK, an enzyme that works like a switch. MEK activates ERK by adding a small phosphate tag, turning it into phosphorylated ERK, or pERK. ERK activity is ERK with that phosphate tag. pERK then travels into the nucleus and turns on activity-related genes. In Fragile X, this process happens too often and is overactive. Now, if MEK is a switch that turns ERK on, blocking MEK should mean ERK gets activated less often. So, can MEK inhibition lower ERK pathway activity in Fragile X mice? That's a question I tested this summer under Dr. Ning Cheng. Hi, my name's Assad, and I'll be briefly walking you through my research, what I discovered, and how it can help advance Fragile X syndrome research. We used the FMR1 knockout mouse, knockout meaning that the FMR1 gene is disabled, which allows the brain to lack FMRP. We focused on the prefrontal cortex because it supports attention, working memory, and self-control, skills often affected in Fragile X. Our MEK inhibitor was PD, a selective MEK blocker investigated in recent studies for regulating brain activity. So hypothetically, PD should reduce ERK activation in Fragile X mice while total ERK stays the same in both sexes. We'd know it worked if the pERK bands in our Western blots, protein quantification method, are lighter across samples, with total ERK banding remaining stable. We first treated Fragile X mice by intraperitoneal injection with PD at 1 mg/kg once daily from postnatal day 47 to 51. We chose this late adolescent window because the blood-brain barrier is more mature and the prefrontal cortex is functionally active. We then dissected the brain tissue, homogenized it, and prepared our protein samples. We ran an SDS page to separate proteins by size, transfer those proteins to a membrane, and use a primary antibody to bind to the protein, along with a secondary that binds to the primary and produces light and chemical imaging to visualize our bands. We also did a background correction and normalized bands to a control. Our targets were pERK, total ERK, and beta-actin, which was our loading control. We ran a PD-treated and a non-treated group, with each group having about 7 males and 7 females. Our primary readout was pERK to ERK, where we measured the intensity of the pERK band while measuring the ERK band on the same lane and then dividing to look for activation. Our secondary readouts are our controls for loading and ERK stability. Here are our Western blot images. Actin and total ERK stayed stable across lanes, pERK looks lighter in PD lanes, which matches our prediction, and vehicle also showed slight changes. When we ran statistical tests, our two-tailed unpaired t-test showed no significant difference, with the pattern still fitting our hypothesis that blocking MEK could lower ERK activation. We also wanted to extend the study by looking at significance in sex differences, so we ran a two-way ANOVA and found that PD-treated females had higher pERK ratios compared to PD males, which was significant. However, there was no difference between PD and vehicle overall, meaning ERK activity reduction wasn't confirmed. Now, prior work showed that blocking this pathway can reduce overactive signaling. Our study tests the idea at the molecular level in the PFC and also examines sex differences. We refine what's known by linking a clinically relevant MEK inhibitor to region-specific ERK signaling in Fragile X. The spread in our data could have also hit a real effect. To address this next round, we'll increase mice per sex to boost power and test whether true sex difference exists, add a wild-type baseline to directly compare Fragile X and non-Fragile X mice, and vary dose and timing such as different drug concentrations. Bottom line, we tested whether a drug can calm an overactive brain signal in Fragile X mice. The trend points the right way, and our next steps make the test decisive. Research like this moves us from theory to something we can measure, and hopefully one day treat. Special thanks to my supervisor, Dr. Cheng, and members of the Cheng Lab. This would not have been possible without you all, and thank you so much for watching.